in Seldovia, Alaska, currently, where this project takes place. I work at the Kachemak Bay NERRS with Kui Walker, who is the lead on the project. I'm helping out with the project as well. There's the title. There's a little picture of me, and let's go. So the issue and need we're talking about is that harmful algal blooms, or HABs, are increasingly over the past 10, 20 years becoming of concern in Alaska. They're not new to Alaska, but communities are very concerned about their potential effects on the marine ecosystem and thus marine resources, things people care about. Where this project fits in, which is the Catalyst project, is that we're focusing on a piece of this larger HABs issue where we're looking at paralytic shellfish poisoning toxins generated by HABs. Specifically, we're looking at STX here. And this diagram here is kind of our working understanding of basically the food web and how the toxins are moving through it. Something I'll point out is that the toxins, we understand this really well. We understand them at the phytoplankton level and at the zooplankton level. We're finding them in all the other trophic levels, but can't replicate in the lab because of laboratory reasons. So this Catalyst project is focusing on this next crucial step to understand why we're seeing saxitoxins prevalently, pervasively throughout the marine food web, and try to better understand that. So who are our users? We have a network of folks interested in HABs and their effects. About half of them are interested in the shellfish aspect, so toxins moving through shellfish into people, and that's a human consumption and a health risk. The other half are more interested in the food web ecology aspect, which this project fits under. I'm not going to list all these, but there's a bunch of folks in Alaska working on this issue, this big picture issue in Alaska. This little map is kind of where I am right here, this little dot, and then here's our project partners. And this project is kind of meeting the needs of mostly the food interested folks, but also the Alaskan community. So generally people interested in toxins affecting their ecosystem. How we're doing this, we'll start with the non-engagement piece, because engagement is not fun to visualize that. So we're collecting forage fishes using little beach seines, this method, and we're gonna transport them into tanks. This project is meant to set us up for a more detailed dosing study, and we're doing engagement throughout the project. You can visit the slides. Our products and benefits are, with this Catalyst project, we're going to better understand the feasibility of aquaculturing these forage fish species, which hasn't been done for Alaska species. This project, this 1 year Catalyst, really sets us up nicely for the next research phase, which is the actual dosing and gathering of laboratory information. And that will be interesting overall for this HABs saxitoxin issue, where we can, again, better inform a working ecosystem model of how saxitoxins or PSP toxins are affecting different trophic levels. So the critical piece is kind of moving to that next trophic phase up, which is the fishes. So we can get at some ecologically relevant data to better interpret what other folks are finding in birds, whales, other marine mammals, predator fish, and such.